Hello students, in today's session we'll discuss Hershey and Chase experiment. Hershey and Chase experiment. In the last session we discussed two experiments to prove DNA is a genetic material. One, Griffith transforming principle, and second one is biochemical characterization of transforming principle, or it is commonly known as Everett's experiment. By Everett's experiment, they finally came to conclusion that DNA is a genetic material which enters into the cell and which helps in transforming genetic information. But there was a little confusion whether the protein enters into the cell or DNA enters into the cell. For this, the non-controversial proof that DNA is a genetic material came from Hershey and Chase experiment. Alfred Hershey and Martha Chase in the year 1952. And you already know that the earlier experiments were conducted by using bacteria, whereas Hershey and Chase experiment conducted series of experiments by using T2 bacteriophage. They used two components. One is E. coli cell and the second one is a T2 bacteriophage. And the T2 bacteriophage is a viral particles or viruses that infects E. coli cells or that infect bacterium. And the second, during the course of experiment, they used radioactive substances to identify the movement of molecules or to trace the position of the molecules. So, the T2 bacteriophage has two components. One is a protein coat, or we call it as an outer capsid. The second one is a core DNA. Look at here. The first component is the protein coat or outer capsid, which is made up of protein, and the protein contains sulfur but not phosphorus. And the second component is the core DNA, and the core DNA contains phosphorus but not sulfur. Okay. So when they are allowed to infect E. coli cells by using T2 bacteriophage, what happens during the course of infection? What happens? The E. coli T2 bacteriophage attaches the host cell or E. coli cells. They inject DNA into the host cell, and the host cell DNA treat foreign DNA as a host. Okay, foreign DNA as if its own DNA, and the foreign DNA gets incorporated into the host cell DNA and start multiplying. So after multiplication, the viral particles, the more number of viral particles are produced in the host cell, that is E. coli cells, and those viral particles they come out or ruptures the host cell and they start to infect other cells. So during this experiment, Hershey and Chase, they used two radioactive isotopes. One is P32, phosphorus 32, and the second one is sulfur 35. And they grew different bacteriophages in two different media containing these radioactive isotopes. In the first media, they grew T2 bacteriophage in a media contain sulfur 35, and this sulfur 35 is incorporated into the proteins. You already know that the protein contains sulfur but not phosphorus. So S35 incorporated into the protein capsid, since protein contains sulfur but not phosphorus. Such radioactive labeled bacteriophages are allowed to infect E. coli cells. So during the course of infection, what happens? The protein coat remain outside, whereas the viral DNA penetrates into the host cell, Then E. coli cells are gently agitated to loosen the attached particles, or the viral particles, the attached infected viral particles are separated from E. coli cells by agitating. Soon after agitation, they are centrifuged. Okay, then a culture where now they are centrifuged. The two components are obtained. The heavier components which settle down bottom of the centrifuge and which is called as a pallet, and the lighter one, okay, occupies top of the centrifuge, and that is called supernatant. So in this experiment, the radioactivity, so in this experiment what they observed is the proteins are lighter and bacterial cells are heavier. So proteins are present in the protein coats are present in the supernatant, and radioactivity is detected in the supernatant, and no radioactivity is detected in the bacterial cells. So in the supernatant they exhibit radioactivity, whereas in the bacterial cells you don't find any radioactivity, because proteins do not enter into the bacterial cells. In the second experiment, the other T2 bacteriophages were culture on a medium containing P32. The T2 phases in a medium P32. So P32 incorporated into the DNA, since DNA contains phosphorus but not sulfur. So such T2 bacteriophages were allowed to infect E. coli cells. Okay. The capsid remain outside, whereas DNA, radioactive labeled DNA, penetrates into the cells. Very important. Radioactive labeled DNA, that is P32 DNA, penetrates into the host cell. Then E. coli cells were gently agitated to loosen the attached particles. Okay. Then the culture were centrifuged. The two components are obtained, supernatant and pallet. The supernatant contains protein coats, whereas the pallet contains bacterial cells. When they analyzed the centrifuged, they could able to identify the radioactivities found in the pallet. No radioactivity is seen in the supernatant. So it finally they came to conclusion that the DNA enters into the host cell, and it acts as a genetic material that passes from virus to bacteria. So look at the picture. So two different experiments were conducted. So in the first experiment, they grew T2 bacteriophage contained sulfur 35, and in the second experiment they grew T2 bacteriophage in the P32, and they are allowed to infect. Okay. Finally they could able to identify supernatant and pallet. In the first experiment, sulfur is detected in the supernatant, and no sulfur detected in the cell or pellet. In the second experiment, phosphor detected in the cells or the radioactive phosphorus is detected in the pellet. No radioactivity is found in the supernatant. So Hershey and Chase, they concluded that the viral DNA carries the information for production of more viral particles. So DNA is a genetic material that passes from virus to bacteria. Okay. Next, we'll move on to the properties of genetic material. You already know that DNA and RNA are the two genetic material. DNA is a genetic material. DNA is genetic material found in the most of the living organism, whereas RNA acts as genetic material only in few viruses. What are the properties? The genetic material must have the following properties. The first one, it should be able to produce its replica. So both the DNA and RNA produce its replica, whereas proteins will not produce its replica itself. So that's why DNA and RNA act as they are considered as a genetic material. Second one, it should be chemically and structurally stable. Third one, it should scope for slow changes that are required for evolution. So usually the genetic material undergo mutations, and mutations are the raw material for evolution that leads to origin of new species. And fourth one is it should able to express itself in the Mendelian characters. Okay, look at here, both nucleic acid DNA and RNA can replicate but not proteins. So that's why DNA and RNA are considered as a genetic material. When you talk about DNA versus RNA, when you compare DNA and RNA, the most stable genetic material is DNA as compared to the RNA. Why? Because the first one, the presence of thymine at the place of uracil confers the stability to DNA. So you already know that nitrogen bases of DNA, one is adenine, guanine, cytosine and thymine, and the nitrogen bases in case of RNA, adenine, guanine, cytosine and uracil. So in DNA thymine is present, in RNA uracil. Instead of thymine, uracil is present. So thymine confers the stability to the DNA, the first reason. The second one, so both the DNA and RNA are able to mutate. Both have a capacity to mutate. When you compare DNA and RNA, RNA being unstable mutate at a faster rate. Okay. So RNA mutates faster rate, whereas DNA mutates at a slower rate. Okay. That's why it is stable genetic material. Okay. The question arises, why RNA is unstable? Okay. Because DNA is chemically less reactive and more stable as compared to the RNA. The question arises, why DNA is less reactive, chemically less reactive and structurally more stable? Because in case of RNA, each nucleotide has two functional groups, that is two hydroxyl groups. Okay. If any molecule has more number of functional, more reactive and highly unstable, whereas in case of DNA, nucleotide of DNA consists of only one functional group. That's why it is a less reactive and more stable. So when you compare DNA and RNA, so DNA is a more stable genetic material than the RNA. So in this session, I will complete this session. In the next session, we will discuss the mechanism of replication. Thank you.